Like we stated earlier in the lecture, the quantitative relationship between the physiological responses and the concentration of the agonist can be measured by using bioassays. The first part of the drug-receptor interaction, which is the binding of drug to receptor, can be studied in isolation using binding assays. And the localization of receptors can be performed via radiographic studies. First up, let's discuss bioassays. Bioassays involve the use of a biological tissue to relate drug concentration to a physiological response. Usually, isolated tissues are used because it is then easier to control the drug concentration around the tissue and reflex responses are abolished. However, bioassays sometimes involve whole animals, and the same principles are used in clinical trials. Bioassays can be used to estimate the concentration of a drug, largely superseded by chemical methods, its binding constants, or its potency relative to another drug. Measurement of the relative potencies of a series of agonists on different tissues has been one of the main ways used to classify receptors, like adrenoceptors. Then we have binding assays. Binding assays are simple and very adaptable. Membrane fragments from homogenized tissues are incubated with radiolabeled drug, usually tritium or 3H labeled, and then recovered by filtration. After correction for nonspecific binding, the 3H drug bound to the receptors can be determined and estimations made of Ka and Bmax. Number of binding sites. Binding assays are widely used to study drug receptors, but have the disadvantage that no functional response is measured, and often the radiolabeled drug does not bind to a single class of receptor. Now, how can we perform the localization of receptors? The distribution of receptors, for an example in sections of the brain, can be studied using autoradiography. In humans, positron-emitting drugs can sometimes be used to obtain images in the case of positron emission tomography or PET scanning. They show the location and density of receptors, like dopamine receptors in the brain. So, this was all about the important points to remember about drug-receptor interactions. Leave us a comment to continue the discussion. Stay tuned for more amazing videos only on sqadia.com.